Imagine, if you will, a tiny soil nematode, just a microscopic worm, wriggling through the damp, unseen world right beneath our feet.
Okay.
Its head passes through this minuscule loop. It looks like nothing, just a bit of fungal hyphae. Then its body slides through, smooth as anything. But just as it's about to clear the loop, bam, disaster. That loop suddenly inflates inward, grips the worm tight. It
Wow.
thrashes, struggles, but nope, it cannot escape. It's caught. Doomed, actually, by a fungus.
Yeah.
And when we typically think of fungi, we picture, well, gentle decomposers, silently breaking down matter. But what if I told you some fungi are secret predators, setting these ingenious traps, even launching microscopic harpoons at tiny animals?
It's really something, isn't it? What's truly insightful here is just how far fungi have pushed the boundaries of their lifestyle. We often think of them as, slow growers, stealthy permeators.
Right, just kind of spreading out.
Exactly. But these highly evolved hunting strategies show this dynamic, aggressive side. It's not just about breaking down stuff. It's about actively capturing and consuming. And it's fundamentally driven by the need for a crucial resource: nitrogen.
Ah, nitrogen. Okay.
Yeah, these battles play out in the microcosms of soil, compost piles, rotting logs, wherever these tiny animals are, representing a really valuable protein supplement.
Like a protein shade for fungi. And in this deep dive, we're going to explore that astonishing, almost sci-fi world of carnivorous fungi.
It definitely feels sci-fi sometimes.
Totally. We'll focus on how they exploit tiny creatures like nematodes, rotifers, and we'll delve into, get this, 11 distinct and ingenious mechanisms they've evolved.
11. That's quite an arsenal.
Isn't it? From motile spores that actively seek prey to, well, the most sophisticated biological traps you can possibly imagine. So this is your shortcut to getting well-informed about a hidden, dynamic ecosystem right under our feet. And trust us, you won't look at fungi the same way again.
I think that's safe to say. And these adaptations, they're found across over 150 species, spanning 5 major fungal phyla: Chytridiomycota, Zygomycota, Ascomycota, Basidiomycota, and even the oomycota.
Wow, across the board then.
Pretty much. It's a testament to the sheer diversity and adaptability of life. Even at that microscopic scale, each mechanism is like a unique solution to the same problem: getting enough nutrients.
Absolutely. Okay, so let's jump straight into these mechanisms. We'll start with the sneaky attackers, fungi that directly infect their prey. First up, motile spores. These are mainly in the Chytridiomycota and oomycota. How do they use movement?
Right. So these fungi deploy cells that have flagella, like tiny whip-like tails, which let them actually swim and seek out their prey.
Actively swim.
Actively swim. Take Catenaria, that's a Chytridiomycota. Its spores have just one flagellum uniflagellate, and they swim directly towards a nematode. They do this using chemotaxis.
Chemotaxis, sensing chemical signals.
Exactly. They sense the chemicals the worm gives off. Once they reach it, they basically form a protective wall, usually near the nematode's mouth or anus. Then they penetrate the cuticle and attack the internal organs. It's pretty precise targeting for a microbe.
Wow, like microscopic guided torpedoes. That's incredibly sophisticated for just a spore. And the adaptability doesn't stop there.
Yeah.
So you mentioned Myzocytium. An oomycota uses biflagellate spores, but it has a backup plan.
It does, yeah. Some Myzocytium species are really efficient. If their spores, the ones with 2 flagella, don't quickly find a host while they're swimming around.
Wasting energy.
Right, they conserve energy, they insist, and then they develop this special adhesive bud, like a little sticky spot. This lets them just stick to any nematode that happens to pass by, so they switch from active pursuit to, well, passive ambush. It shows amazing resourcefulness.
That's clever. Okay, from swimming spores to something even wilder. Injected spores. Specifically, these harpoon cells in the oomycetes genus Haptoglossa. A harpoon? Seriously, how does a fungus make and fire a harpoon?
It's a truly remarkable bit of biological engineering. Imagine this specialized cell. It doesn't move, it sticks to the substrate, and it has what you could picture as a tiny barrel pointing up.
Okay.
This harpoon cell maintains extremely high internal turgor pressure. That's the pressure inside pushing against the cell wall.
Right, like a balloon full of water.
Exactly. And when prey, like a nematode, just brushes against it, there's this built-in line of weakness in the cell wall that ruptures. An internal tube, tipped with a harpoon-like structure, is then rapidly everted.
Diverted, like turned inside out.
Precisely, like one of those party poppers. It shoots out with enough force to actually penetrate the animal's outer layer and inject the fungal genetic material inside.
That's basically a microscopic, high-pressure hypodermic needle. It makes you think of jellyfish stinging cells, the nematocysts.
It really does.
It's amazing that such a complex thing evolved independently in totally unrelated organisms. Convergent evolution right there.
Absolutely. It really highlights how similar environmental challenges can lead to remarkably similar solutions, even across different kingdoms of life. Once inside, Haptoglossa grows through the animal's organs, absorbs nutrients, and, well, the host dies pretty quickly.
Game over.
Yeah. The corpse then becomes a nursery, basically, producing lots of mitosporangia. These make more flagellate spores, or more of those injective harpoon cells to continue the cycle. And in related fungi, like Myzocytium again, sexual reproduction can lead to aldegonia structures full of thick-walled, resting zygotes. They look, well, rather charmingly, like little pea pods.
Pea pods of doom. Okay, from harpoons, let's move to adhesive spores. Sticky projectiles. These fungi make spores designed just to stick to victims. Tell us about Meristacrum.
Right, Meristacrum. It's a zygomycete that goes after nematodes, and it's a great example. It grows these tall structures, brachiophores with twisted tips, and they forcibly shoot out sticky single-spored mitosporangia.
Swoops them out.
Yeah, and what's really clever is if they miss a nematode, they have a backup plan too.
Another one. These fungi are prepared.
They are. The spore can germinate and form a small secondary spore coated in adhesive, sitting on a little stalk, just waiting for another chance. This actually means an infected worm, while it's still moving, can spread the infection to others. It becomes a living vector.
Wow, turning his victim into an unwitting accomplice. Do other fungi use this sticky spore trick?
Oh, yes. Some hyphomycetes, like Verticillium and Maria, they also use sticky spores. We call them conidia in this group. Once their germ tube penetrates the worm, it's quickly, as the text puts it, riddled with assimilative hyphae. Basically, fungal threads fill it up.
It doesn't sound good for the worm.
Not at all. And another interesting one is Nematoctonus leosporus. Its conidia, after detaching, grow this little vertical extension that ends in a sticky infective blob. It's also got unique hyphae with clamp connections, which tells us it's a dikaryotic basidiomycetes anamorph.
Okay, unpack that a bit. Anamorph. Dikaryotic.
Right. Anamorph just means it's the asexual stage of a fungus. Dikaryotic means its cells have two distinct nuclei. And Basidiomytoda, that's the phylum that includes familiar mushrooms. So this Nematoctonus is the asexual form of a gilled fungus, something like Hohenweihelia.
Fascinating connection. All right, the next strategy sounds particularly devious. Ingested spores. The Trojan horse approach. Tricking victims into eating their doom.
Exactly. And Harposporium anglululi is a prime example here. Its conidia are crescent-shaped, moon-shaped, with a sharp point. They're perfectly designed to be eaten by a nematode and then literally stick in the craw, get lodged in its esophagus.
Ouch.
Yeah. From that starting point, the fungal hyphae then grow throughout the host. And what's really intriguing here, connecting to the bigger picture, is that Harposporium anglululi's teleomorph, that's its sexual stage, called Atricordyceps harposporifera, it actually attacks millipedes.
Millipedes, not nematodes.
Right. While the anamorph, the asexual stage we're talking about, targets nematodes. It's one of these rare cases where different life stages exploit completely different animal hosts. Talk about optimizing your resources across different niches.
That is mind-bending. A fungus with a split personality attacking different creatures depending on its life stage. And other Harposporium species also have these edible spores with weird shapes.
They do. H. dicerium has conidia shaped like, well, a tiny high-heeled shoe or maybe a clog. H. rhynchosphorum looks a bit like a cartoon bird without legs.
Seriously.
Seriously. These subtle asymmetries and sharp points aren't just for show. They help the spores lodge perfectly in the worm's mouth or esophagus. Then you have H. helicoides, which has longer conidia. They don't pierce, but they germinate right there in the intestine.
And it's not just nematodes getting tricked into eating these, right?
Correct. Harposporium spirosporum makes these wavy, twisted conidia, sharp at both ends. Rotifers eat them, and they get stuck in their gullet or their mastax.
The mastax. That's the rotifer's grinding organ.
Exactly, they're complex pharynx. Also, at least 12 species of another hyphomycete genus, Bochonia, parasitize rotifers after being ingested. Their conidia aren't pointy, but they still lodge in the mouth or mastax. And interestingly, Paternia species are actually being developed now as biocontrol agents against plant parasitic nematodes, so there's a practical angle too.
From infection to ingestion. Okay, let's switch gears now to the trappers. Fungi that physically snare their prey. You said these traps are either adhesive, using glue, or non-adhesive, using mechanics.
That's right. The simplest adhesive method is probably the adhesive assimilative hyphae, found in some zygomycota like cystipage and stylopage. Here, the entire feeding hyphae are just sticky, covered in glue. Simple but effective, like microscopic flypaper covering the threads.
Just a general stickiness. Okay. From
Yeah.
that, we get a bit more targeted with adhesive side branches.
Yeah, a few species of Dactylella do this. They have specialized side branches coated in glue that stick out from the main hyphae just far enough to snag any nematode that brushes past.
Like little sticky tripwires. Seems like maybe a step up from the all-over glue.
Yeah.
Perhaps an evolutionary precursor to fancier traps.
That's a good way to think about it, yeah. It seems more refined. And Dactylella copepodii uses these branches plus adhesive knobs to capture even larger prey like copepods, tiny crustaceans.
Copepods too. Wow.
Which brings us to the next method, adhesive knobs. These are specialized, swollen cells coated in nematode glue, usually on the ends of short side branches. Found in nearly 20 species across Arthrobotres, Dactylella, and that Nematoctonus we mentioned earlier.
Sticky bumps sound like bad news for a nematode.
Definitely. Sometimes a worm gets stuck to several knobs at once and just can't move.
Yeah.
But what's really clever is sometimes the worm struggles and actually pulls a knob loose. It thinks it's escaping.
Ah, but it's taking the trap with it.
Exactly. The knob stays stuck firmly to the worm's cuticle, and pretty soon it sends out an infective hypha right into the worm. So game over, even if it thought it got away. And some Nematoctonus species, remember? The anamorphs of gilled fungi like Honbohelia, they make these unique hourglass-shaped knobs covered in glue. These ones don't break off, they hold the nematode tight. Even their spores have to germinate and form a sticky knob first before they can infect anything.
So the trap initiating infection is key. Okay, next up, probably the most common trap, adhesive nets. Found in nearly 40 species. These sound complex.
They can be. They're thought to have evolved from repeated anastomosis. That's the fusion of adjacent hyphae, those sticky branches we talked about.
So branches growing, touching, and fusing together.
Right. Early forms might have been just simple loops, but they developed into these complex three-dimensional labyrinths like the ones made by Arthrobotres algospera. That's the most common nematode-trapping hyphomycete out there.
And the complexity isn't just for looks, right? There's an advantage to the 3D structure.
Oh, absolutely. More complex nets are better because they can trap larger nematodes and trap them at multiple points. Makes escape much harder. Interestingly, nematodes often show what's called an aversive reaction. They recoil violently when they touch the net, and sometimes that actually saves them.
A chance for escape.
But if they don't?
If they're caught, an infective hypha penetrates, and the worm usually becomes comatose within about an hour.
An hour? That's fast. Suggests a toxin.
Exactly. It strongly suggests the fungus produces a toxin, some kind of chemical weapon. We'll circle back to toxins specifically. The fungus then digests the prey, and importantly, it moves that energy to its external hyphae to spin new nets and make more conidia. It's very efficient, kind of like a spider managing its web and resources.
Resource management in the microcosm. Incredible. Okay, now for a trap that sounds almost polite. Non-constricting detachable rings. Less aggressive.
It's definitely a more subtle approach.
Sure.
4 species of Arthrobotres and Dactylella make these. A single hypha grows round in a perfect circle, forms a 3-celled ring sitting on a stalk.
When a nematode crawls through, the ring fits snugly, like a collar, and it easily breaks off from its narrow stalk. The worm just keeps going, now wearing its newly acquired collar, as the book says.
Wearing its own doom.
Pretty much. Infection and digestion follow soon after.
So this detachable design, like the detachable knobs or sticky spores, actually helps spread the fungus, right? The victim carries it away before dying.
Precisely. It's a clever dispersal strategy, turning the prey into a temporary vehicle for the pathogen, helps it colonize new territory. And notably, all the species that make these non-constricting rings also make sticky nogs. They often have multiple tricks up their sleeve.
A combined strategy. But now, the main event, maybe? The absolute peak of fungal trap technology. Constricting rings.
The vice grip trap. Yeah, these are the most sophisticated ones we know of, made by about 12 hyclomycetes, especially Arthrobotres and Dactylella. They look similar to the non-constricting ones, 3 cells on a stalk, but the stalk is shorter, stronger. These traps are built to stay put.
And they don't just hold, they actively constrict.
Oh, they do. When a nematode goes through the loop and touches the inside of one or more of those cells, all 3 cells simultaneously inflate inwards. And get this, it happens in about 1/10 of a second.
1/10 of a second. That's incredibly fast.
It is. It grips the nematode in a vice-like grip, basically garroting it.
How on earth does it do that so quickly? No muscles, no nerves? How?
It's all about hydraulics and stored pressure. Those three cells maintain really high turgor pressure, remember? High internal osmotic pressure pushing out. They're held in check by their outer cell walls.
Okay.
Inside, near these subtle lines of weakness, they have folded up reserves of cell wall material and membrane. When the nematode touches the inside, it triggers this rapid sequence.
Yeah.
The outer walls rupture along those weak lines. Water rushes into the cells incredibly quickly over their whole surface.
Because of the osmotic difference.
Exactly. The cells instantly expand inwards into the central gap, unfolding those reserves of wall and membrane, grabbing the nematode. Initially, this expansion actually lowers the osmotic pressure a bit.
But it's already trapped.
It's trapped. And then the fungus quickly pumps the osmotic pressure right back up again, increasing the turgor and effectively strangling the worm. It's an amazing feat of biophysics at the cellular level.
Just astonishing biological machinery. You mentioned they can be triggered just by touch or heat, too.
Yes, even without a nematode, mechanical stimulation or heat can set them off after just a few seconds delay. This whole complex mechanism relies on the ability of eumycotin hyphae, the threads of true fungi, to fuse to anastomose and that rapid pressure release. It's similar in principle to how other fungi forcibly discharge spores, like from an ascus or basidium, or even that harpoon cell we discussed. It shows how a basic physical trick can be adapted for wildly different purposes.
Evolution repurposing mechanisms. Clever.
Very. And they don't just make these traps all the time that would waste energy. They typically only produce them when they detect chemical signals, like ammonia or CO2, that indicate nematodes are nearby.
So they sense their prey is around.
Yes, and some even release chemical attractants to lure the nematodes towards the traps. Talk about setting a deadly ambush. Plus, the spores, the conidia, of these trap formers are usually quite large. They carry enough stored food to build a trap themselves right after germinating, always ready to hunt.
Active hunters, regulating traps, even using lures. Okay, beyond mechanical traps and direct infection, some fungi use chemical warfare, right? Our 11th mechanism.
That's right, toxins. The mycelium, the vegetative part of the common oyster mushroom, Pleurotus astratus, and several related Pleurotus species, secretes a substance that very rapidly inactivates nematodes, paralyzes them.
Just knocks them out.
Pretty much. Then the fungus can easily grow into and colonize their inert bodies. This is really important for fungi like Pleurotus because they often grow on dead wood, which is notoriously low in nitrogen. The nematodes provide that vital nitrogen boost.
Oh, back to the nitrogen again. A key driver. Makes sense. And while nematodes are a big target, these predatory fungi aren't exclusively focused on them.
Not at all. They exploit a wider range: amoebae, rotifers, those incredibly tough tardigrades or water bears, copepods, even little springtails, the columbella.
So quite a menu.
Definitely. For example, Rhopalomyces elegans. It's a striking zygomycete often found on dung. It mostly parasitizes nematode eggs. The eggs release an attractant, the fungus grows towards them, forms these specialized adhesion structures called uppersoria, penetrates the eggshell, and sucks out the contents.
Wow, going after the eggs. Brutal.
It can also parasitize adult nematodes too, and some fungi go for even bigger game, relatively speaking.
Bigger than nematode.
Yeah. Arthrobotrys entomopaga, a hyphomycete, catches the largest known animal prey for any predatory fungus. Tiny springtails, up to about 130 micrometers long. It makes this network of hyphae close to the ground, with clusters of two-celled traps sticking up, each with a big droplet of glue. You can picture a microscopic Gulliver caught by loads of Lilliputian glue traps.
Great image. What about rotifers?
Zoophagous, that's an oomycota, traps rotifers using what the book charmingly calls lethal lollipops.
Lethal lollipops.
Sticky knobs on short stalks. The rotifers try to eat them, get stuck, and the fungus infects them.
Another ingestion trick, sort of. An amoeba. How do you trap an amoeba?
Well, 6 hyphomycetes are known to trap amoebae. There's one really fascinating case with Dactylella passilipaga. Normally, the amoeba Geococcus vulgaris feeds on fungi by puncturing hyphae and sucking out the cytoplasm.
So the amoeba is usually the predator.
Usually. But when it tries that on Dactylella passilipaga, the tables turn. The fungus responds instantly to the attack by rapidly swelling up a bulbous outgrowth right where the amoeba is feeding, essentially gagging it, preventing escape. Then it digests the trapped amoeba.
The fungus fights back and wins. Amazing.
Isn't it? Most amoeba trappers use simpler sticky knobs, though. But it raises a question: why aren't there more amoeba-trapping fungi? Probably it's a matter of scale. A fungus would need to catch an awful lot of tiny amoebae to get a decent energy return.
Makes sense. So with all these incredible, sometimes slightly gruesome mechanisms, you have to wonder about real-world applications, like biocontrol. can we use these fungi?
People have definitely thought about it. Early on, there were suggestions these fungi could control plant parasitic nematodes, which are major agricultural pests.
And did it work?
Well, small-scale lab experiments often looked really promising, but field trials,
Yeah.
generally less successful. It highlights a key ecological point. Putting a biocontrol agent into a complex existing ecosystem is way harder than testing it in a sterile lab dish.
Because the native fungi are already there.
Exactly. Native fungi, competitors, predators of the fungus itself, they often limit the impact of adding more inoculum. The system is already balanced in a way.
Right, the real world is messy. But you said there have been some successes.
Yes, absolutely. Despite the challenges with plant nematodes in fields, controlling problematic nematodes inside the guts of several domestic animals like sheep and cattle has been successfully achieved. That's a significant practical win.
Okay, how do they manage that?
By feeding the animals clamber spores. These are tough, thick-walled resting spores of a particular fungus, Dedingtonia flagrans, which used to be called Arthrobotrys flagrans, usually mixed into their daily feed supplement.
Line of spores.
These spores pass right through the animal's digestive system unharmed. Then they germinate in the dung, in the feces, and produce traps right where the nematode larvae are developing.
Catching the larvae in the dung, breaking the life cycle.
Precisely. It's a really neat practical application of this specialized fungal predation to benefit livestock health.
That's fantastic. What's truly remarkable here, stepping back, is just from microscopic harpoons to these elaborate self-inflating traps, this deep dive has uncovered this hidden world of fungal predation that's so much more dynamic, so much more sophisticated than I think most of us ever imagined. It's not just slow decay, it's active hunting.
That really is the key takeaway. What's so fascinating is the sheer variety of solutions that have evolved to solve one fundamental problem, getting enough nitrogen in nutrient-poor places. These mechanisms just highlight the incredible evolutionary pressures and adaptations happening constantly in the microbial world. They show that fungi, often seen as slow and steady, can also be incredibly swift and cunning hunters. They're masters of their own microecosystems.
We've seen fungi using motile spores to actively seek prey, harpoon cells for pinpoint injections, adhesive spores, and ingested spores for sneaky infiltration. And that whole arsenal of traps, sticky hyphae, sticky knobs, complex adhesive nets, and those just amazing, mechanically sophisticated constricting rings.
Don't forget the chemical warfare.
Right, and the toxins, plus how they exploit all sorts of other microscopic animals, not just nematodes.
We really hope this deep dive has given you a new appreciation for these complex, often totally unseen interactions that shape our ecosystems. So the next time you walk through a forest or even just look at a handful of soil, you'll know there's a silent microscopic hunt going on down there. These tiny, ingenious predators are a vital part of the whole web of life.
Absolutely. And thinking about the intricate design, the rapid action of things like the constricting rings, it leaves you with a question. What other unexpected forms of biological machinery might still be waiting out there to be discovered in the vast, unexplored realms of microbiology? And, maybe more practically, how could understanding these things inspire new biomimetic technologies to help us solve our own challenges?
It's a great thought to end on.
Well, thank you for joining us on this deep dive into the truly carnivorous world of fungi. Keep learning, keep exploring.